lung diseases, cancers, and other things like this in incident prospective setup. In addition to the microbiome data or metagenomic profiles, we have also genomic profiling for most of these individuals, and then we have metabolomic profiling of the blood for a substantial fraction of the individuals. We have exomes, so we can also do some multiomics analysis between these different data sources. So this is the cohort we have been busy with, and National Institute for Health and Welfare collected this, and just 5 years ago, 2016 to 2017, Rob Knight's team currently sequenced the samples for us ultimately. So they were quite long time in storage before sequencing, but the quality, overall quality in the analysis seems to be very feasible. We are mostly focusing on the taxonomic level analysis of this cohort because this is shallow shotgun sequencing, so we can't really go very deep with the functional level, but we can look at the species compositions. And this is the usual principal coordinates analysis figure, and we see here many features that we usually see in population cohorts of the human gut microbiota. So you see here the dominating genus is highlighted with color. I assume you know how to read the PCOA figures, so these are the 7,000 individuals on the map on the left. So we have 7,000 points here, and the proximity of the points on the map shows how similar microbial community composition these individuals have. And indeed, the color is highlighting the dominating genus. We see this usual Prevotella cloud, the green individuals, cloud of green individuals here. That's the Prevotella high individuals. Then we have Bacteroides is the red here, and then we have gradient to other taxa. And this is just something we see in many different population cohorts, including this Finnish cohort now. But one thing you can see here is the group of black points in the middle, and these are individuals who are dominated by members of the Escherichia, and this is something that we see better in the third principal axis, if we would rotate the figure. And we were starting to look at the associations of this of your position on this landscape of the microbiota to different incident diseases, not just the prevalent current cross-sectional disease, but incident disease and future disease risk. And the first thing that we wanted to look at was the associations between microbiota composition and total mortality rates. This is the cumulative mortality rates of the individuals across 15 years after the sample collection, and this is the cumulative deaths on the y-axis. And so far, after 15 years, we have about 10% of the individuals dead, and we can associate this in various ways with microbiota composition. And one of the simplest ways is to take the usual proportional hazards models, and we just looked at the first principal components on the PCOA and BCA. And then you can see that this group of black points, which is visible also in the third dimension a bit better, the Escherichia dominated individuals, they are significantly associated with increased mortality rates 15 years after the sample collection. So we reported these associations between taxonomic composition and increased death risk in the recent paper, which was one of the first summaries of this population cohort. We could also, of course, look at this association in different ways. So in addition to looking at the principal coordinates, we were also looking at taxonomic co-occurrence networks that were coherent. We identified one of the networks that we identified was heavy on these potentially pathogenic bacteria, including again the Escherichia group, but also some other groups that are co-abundant with it. And also the combined abundance of the subnetwork was again associated with mortality rates. And of course, we take compositionality into account. And one nice thing about having the 7,000 samples is we are also able to control a large number of different covariates, including age and medications, cardiovascular parameters, and things like that. So we have enough statistical power. Then we were also looking at specific causes of death, and in addition to the total mortality, just overall mortality. And when we look at the specific causes of death and enterobacterial abundance, which was the driver for this subnetwork, then basically the gastrointestinal deaths are quite significant. This is not very surprising if you look at the gastrointestinal composition and then you look at the death rates. But we also see other types of deaths, like respiratory diseases, cancers, even some neurological causes of death. And these are also associated with the increased death rates. Here you see the list of the different covariates that we included in the model based on the population registers. Then we wanted to see if this association is robust in distinct subpopulations. In Finland, we have traditionally population in east and west a little bit different. They have different genetic composition somewhat, and they also have different cultural habits, including different dietary habits. And so this is good comparison point, because these two populations, subpopulations, are somehow different. So if we can see that the association can be found in both subpopulations, it is of course more robust. And we do see this total mortality association in both of the subpopulations. This is not always the case. For total mortality, we do see this variation, but for other things like liver disease that we have been looking at, the signatures from one region are not always generalizable to other regions. So sometimes, depending on the disease, you may have microbiota disease associations that do not really generalize very well geographically. And this depends always on the disease, but in the case of total mortality, this seems to be quite robust. We are looking at this more in different diseases, also in type 2 diabetes, which is another disease we recently analyzed. We also see this relatively robust geographical replication. Okay, then we were looking at the diabetes. That's another endpoint. We are now starting to look at various different endpoints, and we can use this to define microbiome-based risk signatures, and then we can use this to test the predictions in a leave-out test set. Now lately we have also started to do predictions in different cohorts, to do cross-cohort validations, but we have not published those results yet. But the type 2 diabetes, here you see the usual survival curves for the different risk groups based on the microbiota in the leave-out data, and you can see that the microbiota signature that we associate with increased type 2 diabetes risk is significantly higher in that group. So actually, this seems pretty nice predictions with the type 2 diabetes as well. And of course, then we can analyze taxa in more detail for each disease, but I'm not really going with that. One interesting association is that in most cases, we do see these associations between microbiota composition and incident disease only after 5 to 10 years. So the groups start to separate in the survival curves only after properly after 10 years, usually. So this also indicates that in most population cohort studies where we currently have less than 5 to 10 years follow-ups available, and often smaller sample sizes than 7,000 individuals, we are not able to see these prospective signatures. So thanks to the relatively high sample size and the high follow-up times, we are able to identify these signatures quite reliably. But this is one of the few cohorts with this long follow-up times and comprehensive metadata about the incident disease. Then we have been looking at different ways, different machine learning techniques to optimize the predictions. And there is a lot of work to do in this area in the incident disease prediction. So one of the first things to look at is to compare the different machine learning techniques for feature selection. So we have, for instance, compared gradient boosting to rich and logistic regression, and gradient boosting seems to lead to better predictions. Here, the red bar here shows the liver disease predictions based on conventional risk factors like alcohol consumption and age, BMI and such things, and without microbiota signatures. And we can see that with increasing taxonomic resolution, we can get around the same levels than with conventional risk factors when we try to predict the occurrence of the liver disease. These blue bars are increasing taxonomic resolution, so we go from phylum to class to order, family, genus, and species levels, and then you can see that the predictions improve when we increase the taxonomic resolution. We do not really exceed the conventional risk factors, but of course then the next step is to combine the microbiota signature with the conventional risk factors. And then when we do this, we can indeed get improved predictions. So the conventional risk factors is here. The red one, the risk score from the conventional risk factors, and the blue one here is the risk score from combined conventional risk factors and the microbiota signature. For various diseases, we can improve the predictions by adding the microbiota signatures on top of the conventional risk. So that's one take-home message. And lately we have been thinking more about this feature selection problem in this incident risk prediction, and we are not only looking at microbial abundances, but we are also looking at more phylogeny aware methods. For instance, the phylogeny balances. Some of you might be familiar with this, for instance, the ILR method and other methods that look at the balances between different microbial groups. And we can basically use this to look at any branch of the phylogenetic tree, and we are comparing the relative abundance between the two branches of the tree. And one nice property of this is that it is invariant to the compositional transformation, and we can also deal with various different levels of the tree at the same time, so we don't really have to choose one of the taxonomic levels or do all of them separately, because that's sort of arbitrary. We have been applying this, for instance, in the context of liver disease, and postdoctoral researcher Matti Luuskanen from my team has some quite nice analysis of the liver disease based on this phylogenetically aware feature selection. Okay, so that's thin risk, and we have also been now trying to find different optimal ways of making these predictions, and of course it always may depend on the disease, but maybe we can also say something about the generally optimal ways of doing incident disease prediction in microbiota research. So we organized the Dream Challenge. I have been leading a working group in one machine learning for microbiome cost action, and as part of this cost action we organized the Dream Challenge. I'm not sure if you are familiar with Dream Challenges, but it's a well-known bioinformatics challenge that has covered many different areas of bioinformatics in the last 10 or 15 years. And it's kind of a Kaggle type competition where you can propose a competition. There is usually a prize also available, and then different competitors can propose competing solutions to the problem. So our problem was basically how to predict heart failure based on microbiome composition. And we had some initial evidence from the field risk that microbiome does have predictive power for the heart failure. But we wanted to optimize this, and we set up a challenge. And now the challenge is closed, and we are just writing the manuscript. There were two top scoring teams, and they generated very nice predictions. I think the winning prediction, it's actually not winning, it's top scoring prediction, was based on, I think, just lasso regression and some additional treatment. But for the publication we are now working on, we are optimizing this further together with those top scoring teams. And we can't call them winning teams because at the last moment there were some formal conflicts of interest that we had to take into account. And but they were very highly top scoring teams, so we can't declare the winner in this competition now. But that's another thing, we can still write the publication, and that's hopefully coming out very soon as a preprint. I think we have an ISMB all accepted for this. So if you are going to ISMB, you will hear more about that. Okay, what else? Oh yeah, IHLC also last year they were already telling about the competition. So now one of the new things we are looking at is ARG analysis. Katarina Pärnänen is a postdoctoral researcher in my team. She is now working on this. She already discussed this in some events, conferences. So this is more or less public, but this is our preliminary results from analyzing the ARG composition in gut microbiota and correlating it with different background factors that we have. And we have some significant associations with antibiotic use and sex. One interesting thing is that we are also able to see regional differences, and we are able to see differences related to diet in this cohort, but the further analysis is now ongoing. We have also very nice computing capacity from the Finnish supercomputing clusters that provides those for free to researchers. So we are putting this together now. If you're interested, we can discuss this more. Okay, and then one final thing about the FinRisk is that we are also able to look at multi-omics associations. As I mentioned in the beginning, we have host genomes, host exomes, and host metabolomes for a substantial fraction of these individuals. So we can associate microbiota composition with those things as well. There is one paper that I was co-authoring, not really as a leading author, but it's one of the FinRisk papers. And there is an association analysis between SNP loci and microbiome composition. And they were able to recover a number of associations between the SNP composition and microbiome composition, and to verify some of previously reported associations plus some new ones. For instance, Bifidobacterium levels were associated with LCP locus and variants, which has been linked to lactase persistence in European adults, and that association also differed based on the intake of milk and other dairy products. That's some association that has already been known, but then there are others. There was also the Decalicatenolactaris associated with ABO blood group, and some speculation that maybe it can use the secreted products of the ABO blood group as a food source. Anyway, this paper came out quite recently. You can have more details from there if you're interested in genomic microbiome associations. This is, I think there is more. Okay, this multi-omics combinations is increasingly important. So that's about FinRisk, and then I wanted to do the second part of the talk to discuss a little bit the open data science frameworks that we do for this. And I was running a small course also this week here about this. You know that there are many open data science frameworks available for microbiome research, and this is not an exhaustive list. I'm sure you are familiar with many of these tools if you work with microbiomes. What we have been active with is bioconductor community. And I'm in the bioconductor community advisory board since last year myself, and contributed particularly to microbiome research in the last decade. So I guess you know what bioconductor is. So it is one of the largest bioinformatics open source software development projects in the world. It has been around for more than 20 years now, and there is a steady increase in different contributed packages in bioconductor. And one aspect of bioconductor is that there is heavy emphasis on ensuring the quality of the software. There is software review before you can get the package in, and there is continuous testing and very good standards for this. And there is now more than 2,000 packages available in bioconductor, including microbiome research and other areas like metabolomics, proteomics, and many different sequencing areas. And this is of course a global effort. Here is a map of different users and developers from across the world. It's indeed a genuinely global effort. In last years there has been also more emphasis on multi-omics integration, because bioconductor puts a lot of emphasis on standards for representing different omics data sources. And the multi-omics integration then emphasizes how to bring those together in this bioconductor ecosystem. I'm coming back to this in a moment. For microbiome analysis, there is now more than 100 packages available in R. My colleague Sudarshan Shetty has been listing these bioconductor and R and bioconductor tools on this website. If you are interested, you can find a comprehensive list of all the tools we are aware of in R and bioconductor. This my microbiome R package is one of those, but there are so many more. Many of these have been traditionally based on the PhyloSeq data container that you may be familiar with, and PhyloSeq is a bioconductor data container that is specific for microbiome analysis. But in the last few years, a new ecosystem of tools has started to emerge around more generic data container that we call pre-summarized experiment. And so we can build reproducible workflows by using these data containers, importing data from different standard formats like biome files in R in a certain data structure that I explain in a moment. And then once we have data in our data structure, whether it was PhyloSeq or pre-summarized experiment or other format, then we have ecosystem of tools that we can apply on these data objects in bioconductor. And this has various advantages in terms of interoperability of the tools. We have been starting to build ecosystem around this pre-summarized experiment data container, and it expands the capabilities of the PhyloSeq. In particular, in terms of integrating multiple different data tables and data types. So the starting point for this data container is that you have assay. It's usually a microbial abundance matrix. Microbial features on the rows, and then samples on the columns. And then we can have background information for the columns or samples as a separate thing, background information for the rows. Column background information is usually something for like age and body mass index, health status, living habitat, and so forth. For the samples, the background information for the features could be something like higher level taxonomic classification of the bacterial groups, or living habitats, or some biochemical biotypes. But then we can also have hierarchical background information. For the taxonomic features, it's usually the phylogenetic tree. For samples, it could be nested study designs or host phylogenies or other things. This supports that, and it also supports some other things. We can include multiple different assays that can be different aspects of the microbiome community. I'm coming back to that. Anyway, this is a data container that is compatible with PhyloSeq. It has all the same capabilities and some additional ones. So you can always convert PhyloSeq object into this one, because it's kind of a superset of the PhyloSeq. And then that container that we now use, that has certain beneficial properties. So in addition to integrating various types of site information, we can interlink multiple assays and do this hierarchical data, but it's also optimized in terms of the speed and memory. It supports sparse matrices, which is quite useful for microbiome research. We often have quite sparse data. And importantly, it is integrated with other application areas. So this is a very generic data container. It is not dedicated to microbiome data only, but researchers in single-cell sequencing, transcriptomics, and metabolomics and other fields are using this or very similar data containers. And this way we can utilize the tools developed in these different application domains between these different areas. And then we can benefit from many tools that already exist in other areas in bioconductor. And of course, other researchers who work on those areas can also benefit from the tools that we build for microbiomes. So this makes the tools more interoperable across the whole bioconductor ecosystem, and for microbiome research there is now heavy emphasis on this. So we have importers for the common data formats like biome data and other things. Of course, and then it doesn't really require specific knowledge how you can get your data in this format, and there are good examples available. This is just showing the increase of the different packages in the sequencing studies for bioconductor. This is the bioconductor package number for the different areas of sequencing, and all these are more or less compatible in terms of this data container technologies, and we can cross-utilize the tools. This is a very nice paper about single-cell analysis with a very similar approach. And then there are some online resources for this as well. We have started to build an online book called Orchestrating Microbiome Analysis, and this is something I used also in this course this week. We have compiled a number of examples on how to utilize these tools in the context of microbiome analysis in this online book. It's still beta version and it's developing, but it's very functional. We have been using it in a large number of courses, and then there is similar online tutorials available for single cell and transcriptomics analysis. If you're interested in these areas, they are available online. Okay, I think that's it. Then there are some visualization tools for time series manipulation, time series simulation for microbial community data from many different dynamical models of the microbial communities. And this is an emerging ecosystem. There is certainly more to come, but there is already quite some tools available. And the data integration aspect, as I mentioned, it is increasingly important. So we are not only looking at microbiome data, we are also looking at integration of taxonomic profiles or also metagenomic profiles at the gene or function level to other types of omics. And there are many different levels or hierarchies in this data integration. So we can integrate cohorts, do meta-analysis, but we can also combine different omics. Cross-kingdom analysis is increasingly common now, and this framework supports that. The multi-assay experiment data container is something we use for multi-omics integration, and this is the publication. We have not published this container, but we are building tools around this container. So this container is basically allowing you to bind together different omics. There are specialized data containers for genomic data, for instance, that provide nice capabilities for sequence-based datasets in genomics, and then there are different containers for microbiome data. So these container technologies allow you to seamlessly bind these together. Okay, and then there are some free data resources. So maybe you know curated metagenomic data. They are providing almost 100 different data sets from a standardized processing pipeline, and they share those using this same data container technology. So they are supporting the same ecosystem in the curated metagenomic data. We also now have a new interface for EPI Magnify database, where they share open source microbiome data resources. So we have a R package, which is soon in bioconductor hopefully, and can be used to fetch data from Magnify. And then there are data packages including microbiome data. So there is many open source resources available for microbiome now in this supporting this ecosystem of tools. And we basically have, for single omics microbiomes, we have the three summarized experiment container, multi-omics, and multi-assay experiment container. So these are the tools, and then this is the online resource that you can have a look. And we have been running various courses and training events recently, teaching the use of these tools. So this is from Pune, India, where I was in February, as one of those workshops. Okay, So that's it. And then I will spend a moment on discussing some of the time series analysis, which is more on the computational side of things. So as you know, we have these landscape models of microbiome. If we want to look at the population variation of microbiome community composition, we have this idea of landscape, where we may have some more populated regions and some community compositions seem to be more common population than others. And of course, this community composition is always a mixture of different processes. We have migration from the meta community, and we have external factors, external drivers like diet or temperature, other things. But then we also have the interactions within the community, and those interactions often contribute a lot to the complex dynamics of these communities. And the complex dynamics of those communities can be to some extent captured by interaction models. It may not be practically very easy, and I think one of the main limitations in microbiome analysis or longitudinal analysis of the microbiomes is the very limited availability of time series in most systems. But we can still, for some systems, we do have longer time series, and we can also do simulation studies to at least analyze the possible properties of these systems. We have done different studies on interaction models of microbial communities, and this is one simple interaction model of microbial communities with just like 3 members here. We have growth rates and death rates and species abundances. So this is one of the standard models for microbial community dynamics, and we can extend this by adding more species, and if we have a covariance matrix of the species abundance, we can create some block structure in that, and then essentially the different communities that have high internal correlations, they can form like blocks in this matrix and then behave like almost like a single species. Some years ago, with Caroline Faust and Didier Conze, we were analyzing the effect of the interaction structure on the presence of the alternative stable states, but there is also other mechanisms like memory, which I discuss in a moment. So why do we want to analyze these? Microbial communities do exhibit these like population level structures that we have seen in many cohorts, but how does this structure arise? So we are not only interested in how does it look like when we have a look at the signal snapshot in time, we are also interested in knowing how do the individuals travel along the landscape over time. So in short time periods and long time periods, how do you travel along the landscape? And then we would also like to know whether these states that we can see in the landscape model, do they have some degree of resilience? Are they resistant to perturbations? And yes or no, and how often they may switch between the different states, and so forth. And if these transitions have consequences for health, we can also ask if there are early warning signals for shifting between different states of the microbiome. So we have been doing different analyses, analyzing these properties, these dynamical properties of the microbial communities. One thing we did quite recently with Martin Schäfer, which is more like theoretical ecology type of thing, is we analyzed the expected exit times for fluctuating systems that have alternative states. And if you observe the fluctuations of a system within a given stable state, from those fluctuations you are able to estimate the expected exit time from that state. And this is a bit tricky because your expected exit times depends on where you are exactly here. So when the ball is fluctuating in the stable state, the expected exit time is different here than it's here or in the middle. So when the state is in constant fluctuation, we have to take into account these differences. And there is a way to integrate across all these expected exit times at different parts of the stability landscape. And this can give an overall score that you can estimate the expected exit times And we were showing that this expected exit time can be used as a new measure of ecological resilience. One limitation with this is now that we usually do not have sufficiently long time series to apply this on microbial community data, but we are now working with some probabilistic variants that could potentially allow us to combine information from multiple shorter time series to do something similar. Okay. And then the probabilistic early warning signals is another topic I wanted to mention. So Ville Laitinen, who is in my team and will hopefully defend his dissertation soon this year, he has been studying probabilistic early warning signals. And the concept of early warning signals has been around already for quite some time. And this is the idea that complex systems, when they fluctuate, if they are approaching a state of collapse, for instance, here we just have a simulated example where system is basically collapsing from high abundant states to low abundant state or extinction. And basically in these early warning signals, you think that system fluctuations change before such collapse. So complex system behavior changes before the state shift. And we can measure this change in the behavior. And classical measures for this include things like increased variance and increased autocorrelation, and some other measures like flickering between the states and slowing down. The system slows down, the fluctuations slow down often before the collapse. These kind of things. And you can measure these. This is autocorrelation over time, autocorrelation between consecutive time points over time here at the bottom figure. And you can see at least there is some mild increase before the collapse to the other state, already before the collapse. So these have been based on pretty classical statistics, and these have been reported in many different real systems, including coral reefs and climate and stock markets and so forth. So there is evidence this works in different systems. And autoregressive processes is one of the well-performing signals, and we basically did Bayesian extension of this. So we made a Bayesian formulation of this autoregressive process where we regulate the solution with some Gaussian process smoothing for the parameters. So we want that the parameters of the model do not change too drastically between consecutive time points, and this helps to regularize the model. And then we were comparing this to the earlier approaches, and here is the autoregressive model and time-varying autoregressive models, and then the probabilistic variant. And the probabilistic variant seems to be more robust. This is the posterior estimates from the model, and it's more robust, and it's also a bit easier to estimate the kind of significances or importances for the model. For the classical models, we have to use surrogate models, so we have to take some bootstrapping or downsampling of the data, and we have to relearn the model several times, and then we have to make predictions based on a large number of alternative models that have been learned from different variations of the data, and then we can build some confidence intervals. That's a bit complicated process and a bit heuristic. With probabilistic technique, we can just calculate the posterior probabilities, and we can do the simulations. We can get smoother estimates. And when we compare the outcomes from the model, we can also compare the false positive and false negative rates between these different techniques, and the optimal compromise between the false positives and false negatives is given by the probabilistic model, at least in our experiments. So there are different advantages of this, and we just wanted to have a look if we are able to improve the predictions by building a probabilistic variant of the classical early warning models. This is for univariate early warning signals, and there is now some follow-up work that we published recently on multivariate extension of the same, when there are multiple correlated signals. And I promised to leave some time in the end for discussion, so I will just briefly mention that one thing we have recently been working on quite a lot in these complex systems and time series is ecological memory. And ecological memory is an aspect of a complex system that is quite often ignored in the dynamical models. Most of the dynamical models we commonly use just assume that there is no memory in the system, but in ecological systems there is many different sources possible memory. Species adaptation is one source of memory in the system. Environment modification by species could be another. There are other mechanisms that would explain how memory arises in the system. Also, antimicrobial resistance is to some extent an example of how a system collects memory. Like when antibiotics are removed, the system will still maintain this at least for some time. So there are different mechanisms of memory. And one way of incorporating this into models is fractional calculus. So fractional calculus is something where you use non-integer derivatives in the differential equation models. And this has been shown to have an interpretation as memory. Sorry for this. So fractional derivatives can be interpreted as adding memory in the system. And then we can formulate these time series models from the different alternate dynamical systems by actually modifying them so that we include the non-integer derivatives instead of the usual integer derivatives. And then we can have memory in the system. And we have done this incorporation for different dynamical models, and then we analyze the system under perturbation, and then we can see that what kind of impact the memory has on the resilience. So in the short run, memory can increase the resilience because it sort of prevents the system from moving too much immediately. But in the longer run, memory also makes the system remember the movements, and this way, in the long run, it can be less beneficial for the resilience of the system. So it can also depend on the time scale that we are looking at. It can prevent state shifts in the small perturbations, but in large perturbations, it can again be converse, and then it can also be increasing the oscillations of the system before state shift. And then this way it may support the long-lasting transient states, for instance. Okay, we also showed it can be a mechanism, one new mechanism for forming the alternative states in the communities. This is more theoretical analysis so far, but memory is one of the mechanisms that can contribute to having these landscape models of the microbial communities. I think that's it. We have implemented a simulation for the fractional differential equations in a new Julia package. So if you're interested in Julia, we are also now using some new tools in Julia, not only Python. Especially for these time series models and longitudinal models, sometimes this is important for computational efficiency reasons. And then, of course, acknowledgements. Here is a short list of some people who played key roles in different studies that I was showing today. And these are external collaborators. Then my own team you can find from this page, and they are all important for this research. So this is our website, and all the key players from my team who contributed to this study are listed there. And University of Turku is basically located in southwestern Finland. It was the first academic center in Finland. Our first university was in Turku before Russians invaded Finland and moved that university to Helsinki. And it's now the current University of Helsinki, and we got that close to you where I'm now. So thank you. This was my short talk today. Thank you very much.
Thanks, Leo. And we have time for some questions. I have one to start off. So with the green challenge, did anybody try changing the bioinformatics pipeline?